Hello and welcome to Wannick Science. This video is about the dihybrid cross. I want you to realize that despite the monohybrid cross that we've looked at, the reality is that organisms at large are a complex of thousands or millions of traits, not just one. So a dihybrid cross allows us to examine, of those millions, what are the probabilities of any two traits happening together. So the word dihybrid, di meaning two, hybrid meaning two opposite or different parents. So this is a cross of two different parents looking at two different traits. Here we see two round yellow seeds being crossed. And you can see their genotypes presented there, where round and yellow are both dominant. And you can see that both of these pea seeds are heterozygous for both traits. So if we were to cross those together, what would that look like? As you can see already, the Punnett square is much, much larger in terms of its possible combinations. When we think about the sperm cells that can be produced, we'll just say it's from the one on the right. We need to have one round gene combined with the yellow gene. So we have big R, big Y. We could have little r, big Y. We could have big R, little y. We have little r, little y. That is to say that as alleles are distributed into sex cells, they could come in any of these four combinations. Well, what about the egg cells? Well, it turns out the exact same possibilities exist because they both have exactly the same genotype. So as we start fertilization, this big R, big Y combined with the big R, big Y of the egg cell is going to produce, oh gosh, big R, big R, big Y, which genotype is going to be round and yellow. Well, what about the next one? Well, if we look at that, it's going to be, let's see, big R, big Y, and that's going to have a phenotype of, got it, round and yellow. Well, what about the next one? Same thing. It's a different genotype, but it's the same phenotype. So continue through. We'll do the first six together. Pause the video and finish the dihybrid cross. Go ahead, pause the video and finish off this dihybrid cross. I'll give you a hint, they're not all yellow or round. Okay, so you've had enough time to complete those. Let's see what you should have come up with. The rest of the dihybrid cross, there should be a total of 16 possibilities here, should have the following genotypes distributed throughout the Punnett square. Well, what does this mean? Just like the Punnett square from the monohybrid cross where there was only four, we're simply just going to look at this and identify what are all the possible combinations. But we're going to have to do a little bit more work involved here. What we're going to think about is what are the possible phenotypes available despite their genotypes. Well, we could have round and yellow, round and green, wrinkled and yellow, or wrinkled and green phenotypes. So let's see, what is the probability or possibilities that the round and the yellow genes assort themselves together based on these random fertilization? Well, if we count them, we should find 9 total round and yellow. Well, how many times does round and green assort together? If you're counting, you should find 3. In the same way, what about wrinkled and yellow? One is dominant and the other is recessive, and that shows up 3 times as well. But how often do we see the wrinkled and green? That is to say the double recessive. Well, that shows up once out of 16. So we see this 9 to 3 to 3 to 1 ratio, where we see the double dominant shows up 9 times out of 16. Dominant for one trait but recessive for the other shows up 3 times. The opposite is true for 3 times, and then the double recessive shows up once. So let's say that this particular cross yields 288 pea seeds. Wow, that's a lot. Well, that's not unusual actually for a plant. So out of 288, how many should I expect to show the double dominant? Well, let's see. That's 9 out of 16 times 288. That gives me a total of 162 total seeds. What if I was thinking about the round versus green? So dominant in in the roundness but recessive in the green. Well, that's 3 out of 16 times 288 is about 54. And as you probably guessed, the wrinkled in yellow also is 54. What about the wrinkled in green, the double recessive? How often should I see the double recessive combined together out of 288 pea seeds? Well, it turns out 18. So what does this tell us? Well, we already know that Mendel taught us about the law of dominance. That is to say that dominant alleles are expressed over the recessive alleles, and only time we see recessive is when the dominant is not actually present. Mendel also taught us about the law of segregation. That is that when alleles are separated to form gametes, they separate away from each other so each gamete gets one of those options. If you are homozygous, then both gametes get the same allele. If you're heterozygous, then it's a 50-50 chance of getting one or the other. And lastly, then, the dihybrid cross demonstrates the law of independent assortment, which Mendel tells us is the idea that alleles, when they assort into gametes, assort randomly. What that really means is that dominant alleles don't find other dominant alleles to be with in gametes. Rather, they just randomly find alleles to be put in. It doesn't matter which ones are connected with which. The problem is, Mendel was able to predict all three of these laws, but he had no idea how this happened. If you've been paying attention, and if you've studied meiosis already, you'll realize that meiosis is the key to all of this. Meiosis allows for those alleles to segregate from each other, and meiosis allows for the independent assortment of those alleles by crossing over. So here's your reflection opportunity. Think of cats. In terms of hair color and hair length, two different genes, these are controlled by the following genes. So we've got the B gene for brown and the recessive for white. We've got S gene for short and little s for long hair. So for each of the three different crosses, determine the probability of each genotype and phenotype for the following dihybrid crosses. So I hope you've seen enough to be able to answer these questions. Good luck.